Hello everybody, እንዴት ናችሁ? ሰላም ናችሁ? ዛሬ እንግዲህ ዩኒት ፎር አንድ ፓርት ነው 11ኛ ክፍል ግሬድ 11 ዩኒት 4 cell cycle or cell cycle which includes cyclic process of cell division ስለሱ ነው brief introduction አለው just it's part of genetics talks about cell cycle of course I've taught you ግን ያስተማርኳችሁ ልጆች 10ኛ ክፍል ስለ cell cycle cell division አስተምራችኋለሁ anyway this is additional when you repeat something it will be retained in the long term memory የሚባል education hack አለ therefore today just ስለ cell cycle what is cell cycle and all the process in the cell cycle we shall talk about that let's without much ado let's go straight into our lecture the cell cycle is the title of this portion and first introduction as introduction let's see this growth this biological process common features of living organisms growth reproduction and also transfer of genetic information you see all these processes እነዚህ ፕሮሰሶች ህይወት ያላቸው ነገሮች አጠቃላይ የሚያሳዩቸው common features ናቸው common ባህሪያት ናቸው growth reproduction and passing on genetic material during reproduction or during cell division and therefore these processes these are all possible because of cell division you see growth is possible because of cell division growth in its biological context how can you define growth in its biological context the biological ጽንሰ ሐሳቡ growth ማለት የሴሎች በቁጥር መብዛት ወይንም የሴሎች በሳይዝ በቮሊዩም ከፍ ማለት ሊሆን ይችላል therefore growth reproduction and transfer of genetic information is impossible without cell division or these processes are possible because of cell division cell cycle cell division and the cells on the path to cell division proceed through a series of precisely timed underline this precisely timed and carefully regulated stages of growth together we call them the cell cycle and therefore when you define cell cycle and define አርጉ ብትባሉ ከዚህ የተሻለ definition ላይኖር ይችላል ማለት ምንድነው cell cycle is a series of precisely timed and carefully regulated stages of growth and division of cells therefore the process of cell cycle the process of cell division through cell cycle is series of precisely timed and carefully regulated biochemical process physiological process or stages of growth and then division together we call them cell cycle if we say as introduction this let us define what is cell cycle again let's see what happens in the cells of course we have already ምን አድርገናል cell cycle define አድርገናል a precisely timed and carefully regulated series of biochemical processes which result in cell division is known as cell cycle you can call it again let us see in detail a little detail the cell cycle is an ordered series of events that involve cell growth you see underline this cell cycle is an ordered series of events that involve cell growth where duplicates its genome synthesizes the other constituent of the cell increases in volume by increasing its content including doubling of its genetic material and also its organelles its protein content is multiplied twofold then after it becomes large in size itself forces the cell to divide because when cells become large in size surface area to volume ratio it's a problem therefore becoming large in size is the growth stage then cell division to produce new daughter cells the cell cycle is equal to then cell growth plus cell division these are the two events cell growth plus cell division the cell cycle have to take place in a coordinated way and the events are under genetic control so as to ensure correct division and formation of progeny cells or daughter cells containing intact genome. You see, the process of cell cycle is a very careful biological process. It is well regulated. It is under genetic control. There are genes that work in the production of different types of proteins. These proteins control the process of cell cycle, the whole process of cell cycle. There, I will teach you later when we come to that point. There are cell cycle checkpoints. You see, there are different phases. When the cell moves from one phase to the other phase, there are checkpoints. These checkpoints are checking, biochemical checkpoints. They are checking whether this process has been completed before it jumps to the next process of cell cycle. Otherwise, if the cell is not completing its process in the one stage, it will not be allowed to jump to the next stage. አያችሁ አንድ ስቴጆች አሉት ፌዝ የሚባል አለ ሴል ሳይክል ውስጥ አንድ ፌዝ ላይ ማጠናቀቅ ያለበትን የስራውን ሳያጠናቀቅ ወደ ሌላው ጀምፕ ማድረግ አለበት ቼክ ፖይንቶች አሉ አያልፍም ይልፍ አይሰጠውም ምን ይሆናል ወይ ሴሉ እንዲጨርስ ለምሳሌ ዲኤንኤው ተሸራርፎ ከሆነ ዲኤንኤውን ሜንቴን እንዲያደርግ ሪፔር እንዲያደርግ ዲኤንኤውን ሪፔር ማድረግ ካልቻለና if the DNA is beyond repair serious damage ከደረሰበት ሴሉ ራሱን በራሱ እንዲያጠፋ ይገደዳል አቤል ከብቶ ዛት ፖይንት ምን ይባላል ይሄ ፕሮግራምድ ሴል ዴስ ሴሉ ራሱን በራሱ ያጠፋ ምክንያቱም የተበላሸ ዲኤንኤ ይዞ የተበላሹ ክሮሞዞሞች ይዞ አብኖርማል ክሮሞዞም ዲኤንኤ ይዞ ወደ ሌላ daughter ሴሎች ይሄን abnormal DNA ማስተላለፍ ማለት that's it's not normal therefore apoptosis ይባላል ይሄ programmed cell death ወይንም ራስን በራስ የሚያጠፉበት ሜካኒዝም አላቸው genetic machinery ያላቸው ራሱ ስለዚህ what can you say the cell cycle have to take place in a coordinated way coordination there is biochemical coordination believe me and the events are under genetic control there are genes when it comes to genes that control the cell cycle p53 ሚሉት ጂን አለ p53 it's the guardian of the genome ይሉታል ይሄ እጅግ በጣም ወሳኝ ጂን ነው ነግራችኋለሁ መጨረሻ ላይ at the end now let's go to the other ስለዚህ ምንድነው የሚያደርገው it's well coordinated and under genetic ለምንድነው under genetic ነው ለምንድነው ያንዳንዱ ሴል ከአንድ ሴል ዲቪዥን የሚገኙ ዳውተር ሴሎች ትክክለኛ ኢንፎርሜሽን ጄኔቲክ ኢንፎርሜሽን ይዘው ማግኘት ያለባቸው otherwise it's not good cell cycle duration before we talk about directly the cell cycle let us talk about cell cycle duration cell cycle duration cell cycle duration is the time that a cell takes to complete one cell cycle and generally a typical human cell in culture divides once in approximately every 24 hours therefore this is general but there are variations general if you are asked what is the general cell cycle duration for human cells it is 24 hours of course however the duration of cell cycle varies significantly by cell type variation in cell cycle let's see variation in cell cycle duration for example rapidly dividing cells for example embryo cells are one group of rapidly developing cells early embryo cells can complete cycles in 90 minutes alternating only between S and M phase you see they are not going to the embryonic development embryonic growth embryonic cell cycle alternates between S just simply it doubles its DNA and then go to M phase there is no G1 phase and G2 phase that means the cell is dividing without growing significant growth in size is not observed because it's not doubling its materials and therefore that's why it takes very fewer duration of time if you go to again intestinal cells where there is high turnover ዳይጀስቲቭ ሲስተም ውስጥ ሄዱ በየቀኑ የሚሞቱ እነሱን ለመተካት ደግሞ ስቴም ሴሎች አሉ በየቀኑ ሴል ዲቪዥን ይካሄዳል ከፍተኛ ተርኖቨር ያለበት ጂአይ ትራክት ነው ስሞል ኢንተስታይን ላርጅ ኢንተስታይን ምክንያቱም ከፍተኛ ፍጭት የሚደረግበት ነው ኤንዛይሞች አሉ ፉድ ኬሚካልስ አሉ therefore እዛ በየቀኑ የሚሞቱ in millions if not billions of cells እንደዛው ደግሞ የሚተኩ አሉ ከፍተኛ ሴል cycle or cell reproduction ያለው እዛ intestinal cells can divide in 9 to 10 hours 9 to 10 hours slow dividing cells የሚባሉ ማለት slow dividing cells some specialized human cells such as neutrophils እነዚህ ነጭ ደም ሴሎች ናቸው special types of white blood cells neutrophils they take around 12 hours while basophils የሚባሉት ቀናቶችም ይወስዳሉ አንድ ባሶፊል ሁለት ለመሆን ቀናቶችም ይወስዳሉ variation አለ ማለት ነው there are variations now ስለምን አወራን እስካሁን introductory part አወራ cell division without cell division growth አይቻልም reproduction አይቻልም genetic information ማስተላለፍ አይቻልም cell division ከዛ ምን አለ cell cycle define አደረገ precisely timed and well coordinated well controlled genetic program ያለው process that leads to cell division እና cell cycle cell growth እና cell division የሚያካትት process ነው አሁን ፌዞቹን እኔ let's see phases cell cycle ፌዞች አሉት phases the cell cycle has two major phases መጀመሪያ በሁለት ነው የምከፍለው two major phases የመጀመሪያው phase interphase ነው ይሄንን ነው preparatory phase የሚሉት ይሄንን ነው እንግዲህ cell growth ብለን የምንጠራው ሴሉ በሳይዝ የሚያደርገው መቼ ላይ ነው interphase ላይ ነው ትንሽ የሚጢጢ ሴል very small cell becomes big cell during interphase it is preparatory phase እዚህ ውስጥ ነው DNA የሚበዛው ማነው ኢነርጂ ፕሮዲዩስ የሚያደርግና የሚዘጋጀው እዚህ ውስጥ ነው ኦርጋኖሎቹን ሁሉ የሚያበዛው እዚህ ውስጥ ነው enzymes or other protein factors are made and ready before it enters into the mitotic phase or M phase now the interphase እንደገና ስንመጣ interphase ምን ይባላል ብለናል preparatory phase ይባላል preparatory phase ነው መዘጋጃ ነው ለምን ነው የሚዘጋጀው just ለሴል ዲቪዥን ነው የሚዘጋጀው ሁለት ቦታ ለመከፈል መጀመሪያ ይዘጋጃል ምን በማድረግ ይዘጋጃል as I have told you by increasing its cellular materials ሴል ውስጥ ያሉ ማቴሪያሎችን በማብዛት ምን DNA ደብል ያደርጋል DNA አማውንት ይጨምራል ኦርጋኖሎች ይበዛሉ proteinoች energy production preparation the interphase though called the resting phase ይሉታል divide የሚያደርግበት phase ስላልሆነ resting phase ይሉታል ግን strictly speaking rest አላደርግም metabolic active ነው though resting phase the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner ስለዚህ resting phase የሚለው mislead እንዳያረጋችሁ ምንም ነገር አይሰራም ማለት አይደለም ሴሉ እጅግ በጣም ዳይናሚክ ነው undergoing growth busy ነው just you consider as a very busy city the cell is preparing materials including doubling DNA in this case otherwise it is usually called known as resting phase divide ስለማያደርግ resting phase ይሉታል then therefore it is preparation for division by undergoing both cell division and DNA replication interphase is the period of preparation for a cell to divide and during interphase the cell undergoes what these changes normal growth processes gathers nutrients and energy makes copy of its DNA and prepares for the cell division phase M phase and let me give you some remark in this regard you can see here small cell becomes big cell interphase as a cell grows and its size increases there will be decrease in what surface area to volume ratio. I have told you earlier, this is a problem. If you are asked why are cells made small, why nature made cells small, very small, microscopic, just to increase surface area to volume ratio. For what purpose? For exchange of materials. You see, as the cell grows from small cell size to large cell size, you know that surface area to volume ratio becomes decreased. Then what is the problem? Surface area to volume ratio decrease means a decrease in surface area to volume ratio means very slow exchange of material between the cell and its environment, which is not good for the cell. The cell will not give out its products and its waste as fast as they are produced inside, and at the same time the cell will not receive material from the outside like oxygen and nutrients as fast as it needs inside. That's the problem. This can kill the cell. And the solution for the problem is division. Large cell, when it divides into two smaller cells, each smaller cell will have sufficient surface area to volume ratio and a very good give and take between its environment and the inside of the cell. You see, that means exchange of material will be fast, rapid, and no problem. It is significant to note that in the 24 hours average duration of cell cycle of human cell, the division phase lasts for only about 1 hour. Imagine, interphase and M phase or division phase we have said. Division phase or M phase only out of the 24 hours only takes 1 hour. What percent is 1 hour? What percent is 1 hour out of 24 hours? If you consider 24 hours is 100%, what will be 1 hour? Just dividing by 24. By dividing by 24, that means you can even put it, I think more than 90% of the time taken for a cell cycle in human, average cell cycle in human average cell duration, cell cycle duration in humans is taken up by interphase. Interphase takes more than 90% of the time. The interphase lasts more than 95. Oh, I said 95. More than 95% of duration of the cell cycle. The interphase is further. Now we have said about interphase. What is interphase? Division. Preparatory phase. It prepares. The cell prepares. How? By growing in size. The interphase is further divided into three phases. One is G1 phase or gap 1, first gap stage, we call it also. The second is S phase or synthesis phase, and the third G2 phase or gap 2, second gap phase. Let's see what these three altogether take up 95% of the time for the cell cycle duration. Let us see each. Key cell cycle phases. Phase durations, key cell cycle phase durations, they say 24, by taking an average human cell taking up 24 hours mode. Phase 1, G1 takes 11 hours. S phase, synthesis phase, DNA is multiplied in this time, 8 hours. And G2 phase or G2 phase or second growth phase takes 4 hours. You see, we are dividing those 95% of the time for the interphase, the specific. Therefore, out of the subphases of interphase, G1 phase takes up the lion's share of the time, 11 hours. Now, M phase, mitosis phase, 1 hour, we have already said that. G1, let's see G1 phase. What happens? G1 phase is the first growth phase. After completing mitosis and to prepare for the next division. The cell completes mitosis, then if it is going to proceed division, G1, it enters into G1 phase. During G1 phase, the cell is metabolically active and continuously grows, but doesn't replicate its DNA. The cell accumulates the building blocks of DNA. In this time, of course, building block, because after G1 there is S phase where DNA is synthesized, doubled. In order to double DNA, you need raw material for DNA synthesis, which are nucleotides, building blocks, nucleotides. Therefore, nucleotides are synthesized during the G1 phase, but DNA synthesis do not start at G1. Nucleotides and proteins that are used in S phase, DNA replication, like if you say DNA polymerase is the enzyme that is used to replicate DNA. All those all enzymes that are needed in the S phase during replication are made in the G1 phase. Sufficient energy is also accumulated, made. Therefore, this is major event. Therefore, it is preparation for replication. The cell grows in size, accumulates energy, makes building blocks of DNA, and also enzymes required in S phase are made at the G1 phase, the first growth phase. Remark, it says, some cells in the adult, you see, some cells in the adult animal do not appear to exhibit division. For example, nerve cells, heart cells, many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. You see, occasionally some cells do not totally multiply or divide, or some cells pre-divide when they are needed, may enter into resting state. Therefore, then what can you say about this? After mitosis, they will not come to, they will not come to G1 phase, of course. These cells that do not divide further exit G1 phase, get out of G1 phase and enter into an inactive stage. Now it comes, we call it the quiescent stage, or G0 phase, or even resting phase, you call it. Don't confuse with interphase, also known as the resting phase. This one is when the cell is out of the cell cycle, either permanently or temporarily. They enter into G0 phase. Therefore, this is the remark that I want to put in your mind, please. Therefore, there is G0 there. For some cells, there is G0. And out of these some cells, G0 is forever until they die. Like nerve cells, like heart muscle cells. You see? They are deprived of the cell cycle. They are always. Some cells enter into G0 phase until they are required, until they are chemically stimulated to go into G1, then S phase, then G2 phase. Thank you. Anyway, cells in this stage remain metabolically active, but no longer proliferate or multiply, no longer undergo growth and division, unless they are called upon, and to do so depending on the environment of the organism, of course. Cells enter G0 phase due to what causes cells to enter into G0 phase? Of course, lack of growth factors may cause cells to enter into G0 phase. Nutrient deprivation, absence of nutrients for the cell to proceed into G2S and G1S and G2 may stimulate the cell to enter into G0 phase. Or as a normal part of tissue maintenance, cells may enter into G0 phase. Some cells in the G0 phase can re-enter the cycle upon stimulation. I have told you already this. The second phase is the S phase or synthesis phase. Here, what happens? S or synthesis phase marks the period during which DNA synthesis or replication takes place. Yes, exactly. This phase is a stage in which DNA replication proceeds to form identical pairs of DNA molecules, sister chromatides, that are firmly attached to the at the centromeric region. In this phase, the centrosome duplicates and centrioles develop to help organize cell division. Therefore, you see the duplication of centrosome is also happening in this stage, the S phase also. And in animal cells, during the S phase, DNA duplication begins in the nucleus and the centrioles duplicate in the cytoplasm. Of course, centrioles are found in the cytoplasm. During this time, the amount of DNA per cell doubles. Amount of DNA doubles from 2 capital N into 4 capital N. Doubles. And if the initial amount of DNA is denoted as 2 capital N, then it increases to 4 capital N. But, however, there is no increase in the chromosome number because chromosomes are numbered by their centromer. You see, 2 chromosomes now becoming chromatides are attached at the centromers. We count the centromer, 1 centromer. They fuse at the centromer. We count. The sister chromatides are counted as 1. Therefore, literally speaking, there is no increase in chromosome number because chromosomes are numbered by their centromers. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same. That is counted as 2n. But still, the amount of DNA has doubled. You have to be careful. Now, just like this. Now, these two are individual chromosomes. If they join as the centromer, we count this as 1 chromosome because the centromer is 1. We count chromosome by centromers. Here, 1 centromer, another centromer. You see, these are 2 chromosomes. This is one chromosome with sister chromatides. This is 1 chromosome, no sister chromatides, just 1 chromosome. This is 1 chromosome, sister chromatides. These together, when this multiple amount of DNA, this increases and this increases, this changes into this and this changes into this. Therefore, these are homologous chromosomes, but each with sister chromatides. Homologous chromosomes, these are also homologous chromosomes, but not with sister chromatides, just separate chromosomes. You see? The third phase, G2 phase, second gap. The G2 phase is a stage in which the cell replenishes its energy stores that exhaust during the DNA replication. At S phase and synthesis, synthesizes proteins necessary for chromosome manipulation. That means when the cell enters into M phase, it needs proteins for chromosome division, the spindle fiber proteins, all those enzymes that are used to attach chromosomes with spindle fibers so that they can pull the chromosomes into 2 opposite poles. All preparation for the M phase, that is for the karyokinases, that means division of the nucleus or division of the chromosomes, are made here in the G2 phase. Therefore, G2 phase is preparation for the M phase. In this phase, some cell organelles duplicate and the cytoskeleton disintegrate to provide resources for the mitotic phase. Of course, cytoskeletal elements, there are proteins which serve as skeleton boards and cell. They disintegrate and they are consumed for other purposes. Their amino acids, they are proteins. Cytoskeletons are proteins. They are disintegrated to be made later on, and their resources are used for the stage that comes next, that is the M phase. Therefore, cytoskeletal disintegration is another stage here, another process. After completing the interphase, the cell undergoes either mitotic or, of course, depending on meiotic cell division. Meiotic meiotic cell division or meiosis or mitosis. This is diagrammatic representation of the cell cycle. As I have told you here, this is cell growth, that is G1, S and G2, and this is the M phase. And if the cell is not going to enter into the cell cycle, it goes to G0 phase, where it rests. If called upon to divide, it comes to G1 and the process repeats. It comes to G1, the process repeats. And this is diagrammatic representative of the cell cycle. And thank you very much. Bye bye.